Alright, so today we're going to be doing the second session of conjugation. So we set up our conjugation the other day. So we have our two negative controls, FJ, EC, and the two conjugation plates, FJ plus PDH223 and FJ plus PSA11. So if you remember, we plated them in the center. We put the drop of the mixture in the center. Now they are growing. So we're ready to transfer them into different kinds of plates. So if the conjugation was successful, the Flurobacterium johnsonii mutant should have gained antibiotic resistance, and the motility should be restored depending on where the mutation was. So one of these plasmids is gonna restore the motility, and if the conjugation was successful, both the plasmid will give them antibiotic resistance. So to check that, we have two kinds of plates. The plates we are using are called PY2, and one set of these PY2 plates have erythromycin. So this labeling, the red line indicates that they have erythromycin, and these are plain PY2 plates. So we have one PY2 erythromycin and one PY2 plate for each of our conjugation plates. Now we will transfer this onto the PY2 plates. So this time the streaking is going to be different. We're not going to be doing a streak for isolation. This is called a box streak. So I will show one example. So I'm going to start with the FJ. This is the negative control. This only has Flurobacterium johnsonii mutant. So I'm gonna scrape some of the cells out and put one streak closer to the center. And I'm gonna get rid of that applicator stick, use a new one, and do a second streak, starting around the end of the first streak, and then that's it. So the box should be around the center of the plate, because these Flurobacterium johnsonii have gliding motility. So we're going to be testing their gliding motility. They should have enough space on the agar surface to spread around. So the third streak again starts from the second streak and draw a line. So that's it. So those are the three streaks to make a box streak. So now they will grow on the streak, and if they're able to spread, if they have their motility restored, they will spread around the plate. We will do the same thing on the erythromycin containing plates. Again, scrape some out. First streak, and with a new wooden applicator, second streak, and third streak. So in this, we are not expecting any spreading, because this is our negative control, it's the mutant, and we shouldn't see any growth in the erythromycin containing plates because they don't have erythromycin resistance and there was no conjugation. And for the E. coli as well, we will do the same thing on the both PY2 and PY2 erythromycin plates. Here also, on the erythromycin containing plates, we are not expecting to see any growth, because even though the E. coli have the plasmids, their erythromycin resistance is not expressed when they're in the E. coli. We should see some growth on the PY2 plate for both of them, because there is no antibiotics, there's no restriction for them to grow, but we won't see any spreading. And for the conjugation plates, we will do the same kind of box streaking on these PY2 plates and PY2 erythromycin plates. And if our PDH223 was able to restore motility, we should see spreading on the erythromycin containing plates. On the FJ-PY2 plates, FJ-PSA11 PY2 plates, we might also see E. coli, because there is no antibiotics and E. coli will grow. and erythromycin selects against E. coli, and only the FJ will grow on these plates if they have taken up the plasmid. The same goes to this conjugation. Depending on which plasmid was able to give them the good copy of the mutated gene, we will see spreading or the gliding motility in one of our PY2 erythromycin plates. So after we streak all these, similar kind of box streaking, we're going to be incubating them in 25 Celsius